From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on October 3rd, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 111th episode of Matters Microbial. Many thanks to every listener, viewer, guest, and supporter. It really does matter. My Bio 350 Micronauts at the University of Puget Sound continue their microbial journey. It makes me smile. Each Micronaut has isolated 3 water bottle buddies from their water bottles, which have a variety of forms and colors, as seen here. We went on to do colony PCR to determine the 16S ribosomal RNA sequences of each and confirmed great success in amplification via gel electrophoresis, seen here. Next step is to send off for DNA sequencing and analyze the result to find out who goes there. Also, Micronaut Claire re-swabbed her washed water bottle onto R2A media and once again saw great diversity. This always disturbs students. The diversity includes, as you can see here, what I suspect is antagonism between microbes. It's all about the socio-microbiology. Our marine microbiological safari is going well too. We found plenty of agar-degrading pitters, such as this red colony type seen here. Also, with a little work, we've isolated many luminous bacteria from the mix, as seen here in this short movie. Next step is to identify and analyze the organisms. Now, this kind of safari would be a great outreach experience, and I'm trying to find funding to do this kind of thing after retirement. Fingers and flagella crossed. When I teach a tiny taste of biochemistry in my microbiology course, I remind students of this great quote by Szent-Györgyi. Life is nothing but an electron looking for a place to rest. We humans not just humans, we eukaryotes harvest high-energy electrons from our food carried by NADH and deliver that electron to oxygen, making water. We use this energy differential to create the basic mechanisms of life. Microbes are so much more versatile. Some microbes add and remove electrons from metals such as iron. Our podcast guest today sent me this wonderful banded iron formation sample years ago, seen here. As always, friends, when I talk about issues like this, I will put links in the show notes. Other microbes can use the movement of electrons due to their metabolism to generate electricity, as seen in this video of a microbial fuel cell. It really is all about the electrons. So, it is just wonderful to introduce my longtime friend and colleague, Dr. Jeff Gralnick, to the podcast to talk about the work he and his colleagues have done showing how microbes move electrons around in far more sophisticated ways than our own cells do. Welcome to the Quality Quorum today, Jeff. It's great to have you here.
Thanks, Mark. I'm really happy to be here and looking forward to our conversation.
Would you mind telling everybody a little bit about the banded iron formation that you sent me?
Yeah, so this is a rock from northern Minnesota, from a formation called the Sudan Formation, and an old iron mine called the Sudan Iron Mine that was Minnesota's first iron mine. This mine provided a lot of the iron ore that was used in World War I and World War II to make all sorts of things. But it was closed in the early 1960s, and shortly thereafter became a state park. And so it is a place that you can visit if you happen to be up. It's about 3.5 hours north of the Twin Cities. But if you happen to be around there, you can go down and visit and take either a mine history tour or a physics tour. There's a massive physics lab in the bottom of this mine, where they were shooting shooting neutrinos from Argonne National Lab, either Argonne or Fermilab, I don't know, in Chicago anyway, and detecting them in the bottom of the mine for many years in northern Minnesota. So the iron itself, the iron formation itself, is pretty special because it predates the oxygenation event of planet Earth by 300 million years or so. So it's not just like a few days before cyanobacteria start terraforming the planet, which was around 2.4 billion years ago. But the banded iron formation in Sudan is 2.7 billion years old. And there's not great agreement for even how this iron came to be, how it precipitated, because most of the iron formations on Earth correlate to the time of the oxygenation event. But there are some older ones like the Sudan where it's a bit unclear, and some people have proposed that these were made by microbes, iron oxidizing bacteria that were precipitating the iron. But without a time machine, I'm not sure that we'll ever really know.
Now, this is really interesting to remember that with oxygen around, one redox state of iron is practically insoluble. At a different oxygen level it is, and that's what's responsible for the banded iron formations. And I'll put a link in the show notes.
Yeah. And pH plays an important role in this too. So if your pH is low enough, oxygen won't cause the iron to precipitate in the way that it does at kind of near neutral pH. But yeah, this is a trick for microbes whose energy depends on iron, either to breathe, as we'll talk about in a little bit later, or as an energy source itself. The pH really is important because you're either dealing with a rock as what you're trying to breathe, or you're making a rock as a consequence of your metabolism. And both of those are tricky things.
You know, all of this is very interesting because when I have introductory biology students and I'll ask why it is they need to breathe oxygen, they forget that oxygen genuinely was a poison.
Oh yeah,
it was a product.
It still is, Mark.
Yes. How many repair mechanisms do we have?
I don't know. Not enough.
Yeah. So the point is that it was, and I've said this in my class. Imagine some mutant duckweed that was really good at picking up photons, but it generated cyanide as part of its metabolism, is just growing like wild. That's what happened here. Only it was the cyanobacteria, presumably, that released all this oxygen in the atmosphere. And then we, the survivors, well, everything else, the survivors, begin to use that poison as a place to dump electrons, which is so fascinating. Jeff, I will tell students in my intro classes to hold their breath and raise their hands when it starts to hurt. And when they do, I said, too many electrons.
Yeah. Yep.
And it's true. Yeah. And the other part that I want is a lot of folks that I come to, I'll ask them, what do microbes use iron for? Because they instantly think about hemoglobin in us because they're eukaryocentric, and you and I have talked about that before. But what do microbes use iron for is a great question.
Yeah. I mean, many of them use iron for the same reason that we do, in terms of being cofactors for all sorts of different critical enzymes to get things done metabolically. But some iron, some organisms, like the organism that my lab is mostly focused on, use iron as a respiratory substrate. Iron oxide minerals, but also other forms of insoluble metal oxide minerals.
I just love it because it really is. And this is so hard for me because I had such trouble with chemistry as an undergraduate. And now it's like, forgive me, I breathe it. And thinking about what oxygen's used for and thinking about different redox states of iron, it's like much more clear to me now. And I didn't get any smarter. It's just learning how to think.
It's, I don't know, it's understanding, it's being curious about the microbe itself and needing to understand the chemistry to understand the microbe, right? That's me too. I mean, I got C's in general chemistry as an undergrad. I did better in organic chemistry because for me that was like solving puzzles once I learned the language. But Um, and in terms of redox chemistry, like this was a deficiency in my prelim during my PhD that a professor who was at Wisconsin at the time named Paul Ludden like really pointed out, and I had
Yeah.
to basically sit down with him and go over a chapter in one of these books to really kind of wrap my head around redox, because it was something that he felt like I didn't have a good enough understanding for. So
Mm.
in my PhD, I didn't work with these bacteria that we do now, but I did think an awful lot about how iron was moving around in the cells and how iron dependent cofactors were being built inside the cell. And so redox is obviously really important for that.
I think this is like really important for listeners and viewers to hear. Just like remembering that pH is the concentration of hydrogen ions, and we tend to forget that and how that relates to marine microbes and all kinds of things.
Everything.
So the other thing that I showed a short video of are these microbial fuel cells, and I'll put a link in the show notes about that. And you have one that isn't operational right now, and I
Yeah,
don't
it's
know if
still. Yeah, I'm not sure how well people will see it, but like, yeah, this is called a mud wasp, and so this is you can buy these on Amazon, and they're fun little things that you can essentially assemble. This has got, I don't know if you can see the sediment on there, but this would have been sediment from Lake Itasca, where we had taught a field microbiology course for many years. From the bottom of that lake. But essentially, you have these two electrodes. So these are carbon felt electrodes. One would be at the very top, which would be the cathode. The other would be below, called the anode. And it's underneath the sediment, where you have the anode place that essentially is connected to the cathode that's in contact with the air above at the very top. And bacteria down at the bottom can put electrons onto the anode substrate, and those electrons can flow through the system and eventually go to oxygen up at the cathode, which then can make this little light blink, which I know in your video it showed really nice. And that the rate of the blinking is proportional to the flow of current in the system. So the more that the microbes are doing it, the faster it goes. And the Mudwalk guys have a really cool little app that allows you to kind of quantify that over time. It's really nice. And so what these bacteria are doing is they're taking electrons from the food that they eat, and they're moving them to the outside of their cell. And there's lots of different mechanisms that they do this. We understand in Shionella the molecular mechanism to a pretty high level of detail. But there's a whole bunch of other microbes that do this where we understand it to a less degree. And in some cases, we don't understand it at all. Like we'll stare at their genome where we know they can do this, but it doesn't look like it's the same pathway that we would see in something like Shionella or Geobacter or even like some gram-positives that live in our guts like Listeria or Enterococci. There's lots of different pathways to accomplish this, which I think is really neat.
It's all about the electrons and different potentials. And one of the things I talk about a lot in my classes is the fact that I call them the three deadly centrisms. And I'd love to write a little paper about this because it's really true. Students are been hammered into them. They're eukaryocentric, and it's human narcissism. But most life on this planet is microbial and not eukaryotic. Yes, there are some eukaryotic microbes. I got it. Not from you, but other people are thinking that as I say it. Secondarily, we are oxycentric, and we have a very streamlined respiratory complex compared to what microbes can do. And you'll appreciate this, people, even when they're microbiologists, get all carried away with what E. coli does, and I call that coli centricity. Their minds aren't open to the diversity of approaches. Now, you mentioned two bacteria, and I think it's worthwhile, and again, I keep saying this, I don't know where the expression thumbnail sketch comes from, but we use it a lot. Can you talk a little bit about Shionella and then a little bit about Geobacter?
Sure. So Shionella and Geobacter are kind of the main model organisms that people study for this process that we would term extracellular electron transfer. And one of the slides that I provided is are images of Shionella that are expressing green fluorescent protein. And so that's making them glow. So you see lots of kind of little glowing dots. And on one side, they're using iron oxide so that you kind of see this big black chunk in the background. They're breathing iron oxide, and on the right they're breathing manganese oxide. So there you see kind of little kind of more circular chunks, but both are
Mm-hmm.
insoluble metal oxides that are redox active. And so out in ecosystems where oxygen is absent or limiting, like in sediments or in Geobacter's case, say the subsurface, these guys are able to take electrons from the food that they're eating and breathe these insoluble substrates. That has all sorts of interesting functionalities for them, but also really important environmental impacts, because these minerals go from being insoluble to soluble. And so they're mobilizing and changing the redox chemistry of these elements and allowing organisms to now access, say, iron as in its 2 form, or its ferrous form, or manganese in its reduced form, or manganese 2. So they're changing the ecosystems in which they live. For organisms like Geobacter, they are probably doing this almost all the time out in reality. For Shionella, it's actually a pretty close relative to E. coli. They're both gamma proteobacteria. It's a facultative anaerobe in the way that E. coli is. So E. coli knows how to live in your gut, breathing all sorts of different things, but it knows how to live like on your cell phone, breathing oxygen. Shionella is the same in terms of if given the choice, it will prefer oxygen. Geobacter really doesn't like oxygen, and it has mechanisms to kind of detoxify it, but it really doesn't like it. And so one of the reasons, these were organisms that were identified with this capacity back kind of in the late 1980s. By Ken Nielsen and his group for Shionella with manganese oxide as the electron acceptor. And Derek Lovely and his group for iron oxide and Geobacter. So we've known about these for a long time, and Ken tells these really neat stories about how reluctant people were to believe that an organism could breathe something that wasn't soluble, and it took many years to really convince people. That kind of culminated in these works eventually, but now we understand this in lots in like some pretty gory detail, and to the point where we can take most of the Shionella components and put them in E. coli and more or less make E. coli do this, right? So we can reconstruct that pathway. That's not me, but it's other folks in the field. So that helps us really understand like what the fundamental biochemical machinery, what the genes are involved. We still argue a little bit about mechanism, and more so on the Geobacter side than Shionella side, because in Geobacter it's much more complicated for reasons that we don't really understand. So. And the other thing that I would say, even though these things have been studied for going on 30 years, there are still knowledge gaps about how they work. But the other thing is these are independently evolved mechanisms. So the way that Geobacter does things is evolutionarily distinct from how Shionella does it. They do use some common cofactors in their proteins, but the proteins don't seem to be evolutionarily related. And these cofactors are heme groups. So they make multi-heme cytochromes that they are locating in various spots of the cell in order for them to take the electron to the substrate, as opposed to other respiratory mechanisms where the substrate is coming to the electrons. Yeah.
I think that's perfect. And I knew Ken a long time ago, and I heard all those. Who has better stories, by the way?
Yes.
And who has a more interesting shelf in his office? That's what you come down to. He would just hand me stuff and say, "Tell me what that is." He would.
Yeah. Did he also play the banjo for you? He's known for that sometimes as well.
Yes. He plays the banjo. He makes me so angry because he's so skilled at lots of things, but that's okay too. And it's funny because I love talking a little bit about Shionella with students, because they come to me. There's one microbiology course, and they have been told over and over again that microbes are simple, and I actually came up with this great quote. Now, the old, it's ascribed to Monod, but it's not from Monod, as Diane Newman pointed out. Okay, is that what's true for E. coli is true for elephants.
Yeah.
And it was her favorite scientist who came up with that. Who is? We just had his centenary.
Oh, right. Yeah, actually, I have it. I think I have it here. Cliver.
Yeah.
Is that right?
I
Yeah,
never said it
I got the
correctly.
paper here.
Yeah, but the point is, what he said is what, and this idea of comparative biochemistry is important.
Oh yeah.
And I had a recent guest on the podcast who took that quote ascribed to Minot, that was true for E. coli is true for elephants and vice versa. And I have this, and I should probably put it up here. I'll see if I can find the image and make sure you can all see it. And she told me what's true for E. coli is true for E. coli some of the time.
Yeah. I've also kind of espoused some of this. Sometimes I refer to Shigella as the E. coli of the environment, right? And part of that is because Shigella is as easy, if not easier, to grow than E. coli because of its affinity for oxygen and for kind of high nutrient conditions, right? So you can easily reproduce that in the lab. But when you get down into some of the gory details of metabolism, even stuff that we think is true for everything, like there are major differences between how Shigella does things related to TCA cycle and central metabolism, beyond like it's the respiratory capacities, right? Where you have other kind of very glaring differences. But even when you dig down into metabolism, you see very large differences. And what is a challenge that we have in the field is that the way that we interpret genomes of all sorts of different organisms is by and large through the lens of E. coli and our biochemical and genetic understanding of E. coli. And that works to a degree, but it is not as good as everybody would like to think.
And as we learn more, it'll get better. It's
absolutely.
I mean, obviously, it's like when you compare sequences. If you don't have a lot of sequences to compare to, you're not going to know as much as you do. But how do you get there? You have to build on it, and that's something else I want listeners and viewers to understand about science in general. It's iterative and builds on what other people have learned as well. So it's not like
I would say microbiology in particular, like the gaps in knowledge are massive. Even E. coli, the best studied organism on planet Earth, more experiments have been done with E. coli than anything else. About 40% of its genes, we still don't really know what they do.
Isn't that crazy?
Right? It is mind-boggling.
Yeah.
It's the argument for why everyone should be a microbiologist. And even if everyone was, we still wouldn't have all the answers.
Well, and this is why I call myself a microbial centrist, because I am. And I know that several people in the field have tried to think we have to change the way that introductory biology is taught for that reason. Obviously, I think that, but a lot of people don't. So it just takes time. I mean, you know how, and you've heard me make this joke, how science progresses is stage one is that's a silly idea. Stage two, it might work a little bit, but it's not relevant to the natural world. Stage three is I said it was a good idea all along. And one of my research students came up with a great fourth one and I came up with the idea first. We've seen this, you and I, Jeff.
Sure.
Yeah. So anyway, we're off the topic with me flapping my microbial gums. But what I was going to say is this is a good opportunity to talk about some of the projects that you're doing and why they're fascinating to you.
Yeah, so I would say a lot of what my lab has focused on over the years, and this is now my 20th year at University of Minnesota, has been thinking about how extracellular electron transfer, so how microbes get electrons out of the cell, but also how they get electrons into the cell. And this was work that we did in collaboration with Daniel Bond and his group here many years ago, where we took Shigella and tried to make it go backwards. And I think maybe I'll back up one point, and that is say that while minerals are what drove the evolution of these kind of respiratory capacities in the bacteria, we can get rid of the minerals altogether. And this is a field that we will now call microbial electrochemistry or electromicrobiology or whatever. It's got lots of different names. But what really we're able to do is we're able to get the cells, instead of using minerals as their substrate, we can use a poised electrode. And we talked about the mudwatt as an example. In the lab, we have things more sophisticated than that are hooked up to pieces of equipment that allow us to essentially make the redox potential or the surface of an electrode match what the redox potential of some sort of environmental mineral would be. And now the bacteria will use that. And what's really powerful about the electrochemistry is that we are able to monitor those electrons and count them over time and quantify essentially the living activity of those microbes in that system. That allows all sorts of very interesting applications, right? And you as you mentioned, Mark, microbial fuel cells, like this is an application. If you have carbon that you don't care about, and you have the right sort of microbes and the right sort of ecosystem, you can turn some of that carbon into electricity. It's not practical for most of us because we have plugs like in our office or at our home, and the electricity is easy to get and cheap. But say at on the surface of Mars or in the bottom of the ocean, or somewhere where you don't have a plug, like electricity has higher value. But it also is a way that these microbes are natively interfacing with something electric, and so this biotic abiotic interface is powering, no pun intended, a whole neat discipline in something that we would call bioelectronics or biosensing, and where these microbes are being used to sense things. So it's not necessarily the current that is the output of value, but rather it's using current to monitor something, right? Mm-hmm. And there's all sorts of neat applications that's happening in that area. So my lab is focused primarily on the fundamental mechanisms of how Shigella and some of its close relatives. So some Vibrios do this, and some Aeromonas strains do this, but also some more distantly related things that we've grown out of the Sudan iron mine, for example. How they try to do this. But we've also thought a lot about microbes that run the opposite reaction. So in that the second slide that I showed is more of a cartoon of what dissimilatory metal reducers, which we would call things like Shigella and Geobacter, do in terms of their metabolism, where they're eating food and they're taking iron 3 to iron 2. Neutrophilic iron oxidizers take iron 2 to iron 3. And now they're taking that electron and sending it often to oxygen if they're in the right sort of ecosystem, and making a living that way, but they're making rust as a byproduct of their metabolism. Because they're making this insoluble substrate, they also have to do this outside of the cell and bringing the electron in. And one of the reasons, and so I'll give you two examples of microbes that do that. One is called Mary profundus viroxidans. And this has been studied a bunch by Clara Chan, a colleague of mine at Delaware, but also isolated originally by Dave Emerson, who's at the Bigelow research facility. I forget exactly the name of it. But these bacteria are doing kind of the opposite reaction of Shigella and Geobacter, in that they're bringing an electron in. And in that the third diagram that I showed, the third cartoon, is where the electrodes come into play, where things like Shigella and Geobacter, we can use these electrodes to harvest or monitor the activity of these bacteria by looking at current being generated over time, whereas organisms like Mary profundus, or I guess another one is Rhodopseudomonas palustris. Some of these Rhodopseudomonas strains, so Rhodopseudomonas does all sorts of stuff, but some of them are phototrophic iron oxidizing bacteria. And they have a pathway that was initially described by work in Diane Newman's lab, that has some components that are very similar to some of the components that Shigella uses to take electrons out, but Rhodopseudomonas uses it to bring electrons in. And these organisms can interface with an electrode, but as an electron donor or a cathode. And Um, Understanding those mechanisms could pave the way for us to do something that we would call in the field called electrosynthesis, where we use an electrode to power microbial metabolism to, say, fix CO2 and make things for us. Right. And one of the big upsides of that sort of research area is that a lot of biomanufacturing, biotechnology is about how we take plants and process them into something of value. But that's the plants doing the photosynthesis and capturing CO2 and making stuff, right? And plants have evolved to like be very recalcitrant to microbes degrading them. And so that's challenging. In this electrosynthesis model, if we used microbes that knew how to capture CO2 already, but they're now being powered by current from a cathode that could come from a solar panel or could come from nuclear or could come from wherever, right? We could engineer them to make stuff for us. And now we wouldn't have to talk about converting biomass into something else. We could just be making what we need. And so there are some examples of this happening in kind of mixed systems where in many of these instances, the cathode is probably not directly transferring electrons to microbes, but is kind of an inefficient source of hydrogen powering their metabolism in some way. But that's kind of driven my lab to look at microbes in that sphere. The challenge there is that we don't really have like an E. coli of the environment on that side of things. Shigella is very easy to grow. Geobacter is more challenging to grow, but if you know how to do anaerobic microbiology, like you can do it. But for some of these iron oxidizers, like it's really tricky. And unlike Shigella and Geobacter, Mary profundus really only grows like one main way, and that makes it challenging to approach genetically. So one reason we know so much about things like E. coli and Shigella is that they can grow many different ways, which means we can make mutants and pathways that we're interested in and study those pathways because the organism didn't need to grow initially with that metabolism, right? And so, for instance, one of the reasons we know so much about Shigella and its ability to do extracellular electron transfer to minerals and electrodes is that we can make all those mutants aerobically with oxygen where like that metabolism isn't important, right? And then we can test them under these other conditions. Well, for some of these microbes that are really good at neutrophilic iron oxidation, like that's what they do, and that's the only thing they do, and that makes it much more challenging.
Jeff, is it possible to measure what takes place during, I'm going to call it this association between metabolism and electron production in real time?
I talked about in general, like how the electrons from their metabolism are found on the electrode. The fourth figure, which has a little kind of confocal biofilm of Shigella on the electrode in that little bioreactor that we use in the Bond lab. It is so it's highlighting Shigella's anaerobic metabolism, which primarily is converting lactate to acetate, where they're generating ATP through substrate level phosphorylation, and then these electrons have to go through a respiratory pathway somewhere. Otherwise, everything shuts down, as it does when we hold our breath, right? And the graph there is what the technical term is chronoamperometry. But what does that mean? That means that we're measuring current over time, all right? And so current is on the y-axis, and time is on the x-axis. And so over time, what we're counting are those electrons from this reaction that the cells are mediating. We could recapitulate this using iron, but not with the same sort of resolution, right? We'd have to take time points here and again, and we can measure iron 2 over time. And like people do that, we've done that. But one thing that the electrochemistry allows us to do is this real-time measurement of respiration, which is really powerful. So this curve will plateau, and it will continue until the cells run out of food, and then it will fall off a cliff. The magnitude of the current that they produce will be a function of the size of the electrode, right, which is not represented here. So normally when we would put this, we would say like per unit, like per centimeter squared or something, right? If we want to compare what we're getting to, different experiments or to different people. But also it's a function of the amount of biomass on the electrode
Hmm.
surface. So geobacters can generate milliamps of current under this same condition, whereas Shigellellas will generate microamp. And there's two things that drive that. One is geobacters are forming thick biofilms on the electrode surface, and those biofilms are conductive in ways that we argue about a lot. So we don't really know. But this is where nanowires come into play, Mark. You mentioned that word before, and we didn't get back to it. But also, geobacter's metabolism in these systems usually starts with acetate and goes to CO2. And so it is generating 8 electrons for every acetate it consumes, and it is having to work harder for the ATP that it generates. So work harder, manifesting as more electrons on that electrode to generate an equivalent amount of ATP that Shigella generates. And so these organisms do live at different trophic levels. So Shigella slightly above where Geobacter is. And we're in projects that we have been working on in the last couple of years in collaboration with Daniel Bond's lab here at Minnesota. We can tie their metabolism together because of that. So we can have a strain of Geobacter that doesn't know how to eat lactate, but knows how to eat acetate, but a strain of Shigella that is now converting lactate to acetate and feeding Geobacter. And all sorts of neat things happen when they're together that don't happen when they're apart. And that's part of like the baby step at trying to take systems that we can control really well and ask questions of what happens in a community. So when people make these things, they're called benthic microbial fuel cells. So I didn't get into it, but the Navy has funded research in this area for a long time because the Navy has got sensors at the bottom of the ocean that are powered by batteries that they would like to not have to replace, right? And so in theory, something like this, a benthic microbial fuel cell, could power those sensors. It's still hard. So it gets hard in a couple of dimensions. Number one, the bottom of the ocean is freaking cold, right? It's going to be 4 degrees Celsius, and so life is slower there. It's not zero, but it's slower. And then like the food that you might need to power those things sometimes isn't as readily available as you might like. So like the carbon in the ecosystem in the bottom, you pick your random place at the bottom of the ocean, unless there was a dead whale there, like, a few months before, the carbon load is going to be really minuscule. And that's different. Like if you're in a tropical setting, like now it's different because you've got maybe more activity, more things kind of falling all the way to the bottom versus kind of, like real deep middle of the ocean business. Yeah. So, but those, there have been some people thinking about ways to kind of seed that with like, essentially like with wood chips, right? Like let's put a carbon source in there that would
Yeah.
degrade slowly, right? And provide the
Yeah.
power. And there's also more on the electrochemical side of things, which is way like I know enough to like write a grant, but it's not really my area of expertise. But of ways of kind of like stepping up the power and storing the power until you have enough to like perform a function. And so there's avenues there of where you could get more practical applications in these things.
Yeah. Two things real quick. Pete Gurgis, who we've had on the podcast, has been involved with this. And he told me this great story because obviously you don't want to pull up this sensor from the bottom of the ocean to replace batteries. And he said he was really happy with some of the stuff he was getting. And then one of the sensors quit. And so they dragged it up, and a type of a marine snail had drilled into it.
Yeah.
And actually disrupted all the electrodes. I just love the idea of that.
Bioturbation, they call that. Yeah, for sure. Yeah, Pete's a good friend of mine, and we have collaborated in the past on some neat projects looking at kind of looking at genome wise, like distribution of like the Shigella type mechanism. We did in collaboration with him and one of his talented grad students. Pete's fun. One of the things that I love about Pete is I've heard him do this impression twice now. He can give you an impression of what a hydrothermal vent sounds like. Have you ever heard him do this?
No, but
he'll take a microphone and put it real close to his mouth and just like make this noise of like what he says hydrothermal vents sound like. I've seen him do it twice, and it's really funny every time.
so my Pete Gurgis story is he sent me a piece of a hydrothermal vent that I'm sure he wasn't supposed to send me. So I'm all excited. It's about this big. It smells like sulfur. I'm so excited.
Yeah.
And then he texts me saying, just to let you know, you shouldn't probably handle it because of heavy metals and information I could have used.
Funny.
All right. All of this is really interesting, and you're exactly right. Sometimes the most interesting organisms, it's kind of, I kind of view it as my typical luck. The most interesting organisms have the most challenges. And
totally. Yeah.
Yeah, and it's nature resists investigation is what I tell people. But and that's not true. I'm being bitter when I say that, but I have had that experience. But one of the things that I think is interesting is the idea of something biological donating electrons to something that isn't biological. Right. And it makes me think about an idea that was floating around for many years. Well, two things. First, cable bacteria, and also what we used to call nanowires.
Yeah. Well, let's see. Where do we want to start? Because there are those are very different beasts. Let's start with the cable bacteria. And I have the kind of epicenter for cable bacteria is in Aarhus, Denmark, and it's one of the places in Europe that I've been to the most because I consider those folks friends and colleagues. So in I can't talk explicitly about the cable bacteria and how they do what they do, but they noticed almost 20 years ago, maybe 50, about 20 years ago or so, that there was this kind of disconnect in measurements that they were making in sediments, that oxygen was going away in a spot where they expected it to be directly coupled to sulfate reduction but there was a gap. And it turns out this gap is probably being driven by cable bacteria through some fundamentally novel mechanism of conductivity. I think everybody would agree with that statement that I just made.
So we had a guest on the podcast from Austria who had done some work over there. And that's exactly what we're talking about. And what I thought was interesting is the idea of the cells acting like a bucket brigade.
Yeah. Yeah.
And I'm well aware that we don't really know what's going on, which is exciting.
Yeah.
Right? Where I think that the work that you and others have done to show, for example, how some of the cytochromes are able to directly and you've done a lot of work. I mean, I still show a slide from one of your review papers about extracellular shuttles for electrons. Yeah. Because this idea, I mean, we all are used to the idea of breathing oxygen and, an electron acceptor, let's just call it that way. And we have the idea of a snorkel. And if you think about cable bacteria, that's like a snorkel 2 miles long, right?
Yeah, and like, which of the cells in the chain is benefiting energetically? That bit is still a bit confusing to me, and I think they have worked really hard at being able to try to grow these things in the lab, because a lot of the early work was basically done with environmental samples and just trying to understand them in the lab, though, from directly from the environment. And they're getting better at this, and it's still complicated. But the other thing I would say is that so they were able to generate. They've generated tons of genomes now, and if you look really carefully at the genomes, like, we don't see the things that we know are really critical in Geobacter and Shionella. Like, we know that it's something different, right? And that's exciting, but also can be frustrating, right? Because I don't know. I guess I'm more on the excitement. I'm an optimist, right? That means there's things to be discovered.
Oh, yeah. I mean, I say it to students and they don't believe it, but I know you do. It's that I don't know is not a judgment. It's an opportunity.
Yeah, for sure.
And And they can say, well, not on a test. Yeah, I got it. I understand. But that's what I love about the microbial sciences. I'm not put off by things I don't know. They excite me because I wonder how they're being done.
Yeah, it's an opportunity to learn and explore, and that's one of the things that is the most fun about watching people go through that journey in the lab like that. It's really fun. Like that used to be me, right? Knowing what went into the tubes and doing that journey, and now I do what I can to kind of enable that journey for other people.
Yes, yes. So, I mean, I love this idea of an extracellular mediator to move electrons around. So you've already described some of your work with your own microbial fuel cells. You've described electrosynthesis, the ideas that I like so much. What other things would be fun to talk about, do you think?
Well, I don't know. I mean, we could talk more about some of those mechanisms if you want to get into
Sure.
details. We could also talk about one of the ecosystems I'm most interested in exploring at the moment. And again, it's not really me. I mostly do emails and write grants and stuff, but it's my students. So it turns out that like Shewanellas love living in association with fish. And I don't necessarily know why, but we're trying to figure that out. So over the years, when I first got into the field, when I was a postdoc at Caltech back in the day with Diane Newman, I used to be able to read every paper that had the word Shewanella in the abstract. All right, like those days are long gone for all sorts of different reasons. Probably the most important one being that like the field has just exploded, but over the years, but I still try to read all the abstracts. Okay, so again, I used to read all the papers. Now I read all the abstracts, and I noticed this correlation where people who had some fish they were interested in, it could be marine, mostly marine, but sometimes freshwater. If they were interested in the microbes associated with that fish, there was almost always Shewanella in that abstract. And around half of the papers with Shewanella in the abstract are like papers related to fish in some way. So fast forward to a few years ago, where I saw a really cool talk. Karen Gierman, who's a professor at University of Oregon, who has been studying zebrafish microbiomes. Right. So it turns out in her system at University of Oregon, Shewanella was happily there. And so when she came and gave a talk, I was just kind of like on a whim, told one of my postdocs, well, we have in the building next door, you don't even have to go outside, right? Which is important in Minnesota for about half of the year. We have a really nice zebrafish facility that is only used for our teaching labs. And so we went in there and just like did a few samplings, and sure enough, we found a Shewanella in there. And fast forward to more sophisticated experiments where we would actually dissect the gut out of the zebrafish, plate it out, and look for the colonies that were a bit pinkish and sequence 16S. Like we have Shewanella there too. It turns out zebrafish are a powerful model system to do microbiome experiments, because you can take fertilized eggs and treat them a couple of different ways. You can hatch them in sterile water, and they have no microbes associated with them. Sorry, let me be more specific. They have no bacteria or fungi or archaea associated with them. Sometimes they have phage associated with them, and we can talk about that later if you want. But that allows us to do colonization experiments in a really robust way. And I've had undergrads do this. I now have an NSF grant to work on this. But essentially, it is a much more complicated test tube or much more complicated bioreactor that we can use to study Shewanella and other microbes in that ecosystem. And that's some of the stuff that we've been doing for the last kind of 5 or 6 or 7 years, and now with some NSF funding, and hopefully like that's an area that I want to continue to move in. Now, what does that have to do with microbes and making electricity or breathing rocks? I don't know, other than all of the Shewanella isolates that we've tested from these zebrafish, and that's from zebrafish from a bunch of different ecosystems. So we have at the University of Minnesota, we have footprints on the St. Paul campus and the Minneapolis campus. We have another zebrafish facility over in Minneapolis. They also have Shewanella there. One of my postdocs, Andrew Grenfell, who's now an assistant teaching faculty here at Minnesota, was teaching part-time at Carleton College, about 40 minutes south. They have a zebrafish facility. There's Shewanella there too. all of them are able to do metal reduction. All the ones that we've tested anyway. So now, does that mean that's a metabolism that is somehow benefiting them in the zebrafish? I don't know. Maybe that's a metabolism that they really use when they're in between fish, right? When they find themselves in the sediment where oxygen is limited. Maybe. I don't really know, but it's fascinating to think about. And one of the reasons that we started thinking more about the zebrafish actually has to do with these electron shuttles that you had brought up. So Shewanellas secrete flavins all the time. They secrete more when they're oxygen limited, but they secrete them when they're grown aerobically too. And we don't really understand fully why that is, but flavins are vitamin B2. And they're vitamins because we can't make them. All right. So one of the things I've always been curious about is, do these vitamins, so they're redox active, they can take 2 electrons. In the context of metal reduction, we think that helps make different types of iron oxide minerals, quote, look the same to Shewanella. Shewanella has this one primary pathway that gets electrons out, and we think that flavins might help that one pathway be more effective against a wider range of substrates, right? So geobacters don't do this. Geobacters seem to have many different pathways, and so it's way more complicated over there. But that's one of the things we think Shewanella does. But riboflavin is an essential vitamin. You actually can't live without having this. And that goes for everybody, right? There are a bunch of central metabolic proteins that use FMN or FAD, that would be flavin mononucleotide or flavin adenine dinucleotide, as cofactors. And this is not everything makes it, but everything needs it. And not every microbe makes it, but every microbe needs it. And so thinking about one of the things that maybe the fish might benefit from the Shewanella is flavin. And so again, we don't really know anything in that. We know very little in that area, but that was part of the motivation to move to the more complicated test tubes, the much more complicated test tubes that require way more paperwork than I was used to. But here we are.
Yeah.
And, all right. Fun fact for people wanting to get into zebrafish work: that our animal safety folks treat them essentially like mice or rats, and so we have to show that we have valid tetanus shots. Even though, if a fish gives me tetanus, that's got to be a science paper. But anyhow, they treat them the same because they're vertebrates.
So you do know what NASA used to say is that when the weight of the paperwork equaled the weight of the payload, you could launch.
Yes, that makes sense to me. Yeah.
Yeah. And I'm not making light of concerns about animal treatment and safety. I am saying one size seldom fits all, but I understand the drive to make everything under the same umbrella.
Yeah. Totally.
Now, I think this is really interesting, and invariably people want to infer intentionality to associations. And, we don't really know. It's not really how evolution works. So whenever you hear about when I say how evolution works, I mean, Leslie Orgel says evolution's smarter than you are, and it is in that sense. But the idea where you see an association of a microbe to an animal and they must both be getting something out of it, that's tricky. And it's because our perceptions are colored by our experiences.
True. Yeah.
But I love this idea. And the reason that I do is one of the things that's fundamentally different between ourselves and microbes is the limitation of our electron transport chains in us and in our mitochondria, which are, essentially bacteria that have been Darwinoed down to what they are today. But Shewanella has this incredible Swiss Army knife of respiratory apparatus.
Yeah, they hold essentially the world record for most substrates that are breathable for these microbes, right? They know how to breathe lots and lots of different things, and that must give them a fitness advantage in the
Yeah.
ecosystems in which they find themselves. I can tell you a story of one of the Shewanellas isolated a long time ago. They basically this was a group that was interested in the low temperature spoilage of fish, and it's actually the genus Shewanella is named after this person. So Shewan was his name. He was a researcher in the UK, and one of the things they would do is if you just take a dead fish and like put it in a jar and close it up, like you end up getting about a 4 or 5 to the 10, like 10 to the 10 kind of like broth of Shewanella that live on breathing primarily trimethylamine N oxide, but also dimethyl sulfoxide, and they just go to town. And that high nutrient condition with like some weird things to breathe, like they really excel under those conditions. And maybe that's what drives the association with fish, is that when it's time for the fish to go to the great whatever in the sky where fish go, then Shewanella take over, and that's where they go into overdrive, and they sacrifice efficiency of metabolism for speed, and they win in that ecosystem, and then they disperse out into the world to find their next whatever.
It's a great thing to think about. And what I like about a lot of the things that we're talking about with Shewanella is because it's relatively easy to grow, you come up with simple questions and simple experiments, and that's key. Do you remember the old paper by John Beckwith about molecular genetics saying with toothpicks and logic you can solve most problems?
Yeah. I mean, that was part of my pitch for me to even be hired here at Minnesota, was that I was going to be a cheap hire. I needed an autoclave and flames and toothpicks and logic, and like that we could get to where we needed to go. Yeah. A little more complicated now with zebrafish, but again, we have this great facility that in our teaching labs that we have nice access to.
They're a wonderful system for studying developmental biology, and you know I think it's great. So there you go. So I'm watching the time carefully, and I thought you might have a choice here. You can either talk about your route to becoming a microbial scientist, or maybe 3 lessons you'd like everybody to know from today's session.
Let's do the routes. I think people can take away what they please, but I think the route is interesting because very little of what I've done in my career was done preemptively or on purpose. Like it was more or less a curiosity, a fascination, a chance encounter, and I don't know. I guess realizing that there was an opportunity and going after it. So my fascination with science didn't begin with microbes, although it is there now. It began in a high school AP biology class with the lac operon. So there's microbiology in there, but it was this fascination of the logic in a very simple genetic circuit. That would influence or control to some degree, right, behavior and metabolism. And that genetic bit took me to major in molecular cellular developmental biology at University of Colorado in Boulder. And back at that time, this was before Norm Pace was there. It was before a whole bunch of people were there. So there were like the microbiology was like people doing metabolism in yeast, basically. So there wasn't a ton of micro there, but if you were interested in biology, you either did little stuff or big stuff. And so I did the little stuff, the molecular stuff, right? But I then had a very random chance to get into a lab to start doing research. And this was in Drosophila, in fruit flies. And I was doing some molecular biology to try to track down the cause of a wing margin kind of morphogenesis defect that caused the Drosophila to make only half of their wing, essentially. And it was just, again, it was more molecular biology, but I was like, I really liked being in the lab. And all of a sudden, like here I was doing science and understanding understanding science in a way that I wasn't learning in any of kind of the cookbook labs or in any of the courses, right? It was very different. And so I kind of fortuitously found some different summer programs to apply to, and I got into one, and I worked with George Weinstock at then it was the UT Health Science Center. And in George's lab, he had me work with a postdoc named Chris McKenzie, who was a Scottish researcher who I think is still around down there teaching, and had become like super passionate about microbiology. And so once I could understand his very heavy Scottish accent, Chris would share with me. Like every couple of days, he'd give me another paper about a cool microbe, and like Deinococcus radiodurans sticks in my head of like a microbe that could resist like massive amounts of radiation. And I was like, holy, wow, this is really cool. So it was learning things like that. And we were doing some of the early sequencing in bacterial genomes in George's lab, mostly E. coli, mostly cloning, but so sequencing is another thing that's been really ratcheted in my head in terms of the power to understand microbiology through the lens of sequencing. And like my lab doesn't develop any of those methods, but like we certainly use them and think about them. And it's been amazing to see the progression of science from the late 90s when I was in this lab, in George's lab, where it was about $1 per base pair to like sequence DNA. All right. And so that would mean sequencing something like E. coli is about a $5 million project. Yep. And these days we can do it for about $60. And with the newest technology, some of which we have here at University of Minnesota in our genome sequencing core, we can do it for $5 a genome. So like you were talking in the intro, Mark, about sequencing the 16S ribosomal RNA gene, that's going to cost you like $5 or so, right?
That's correct.
We are near the place where like you can just do the genome for $5. Now the analysis is very different, right?
Yes.
But it's we're almost to the point where sequencing is free, and that like is really that will change microbiology yet again. It has changed microbiology across the spectrum as it continues to get cheaper and cheaper, but free changes the game again too. So that really ratcheted in my experience there. From there, I knew I wanted to go to grad school. A wonderful colleague named Adam Drix, who was at the time at University of Chicago, or University of Loyola in Chicago, he was the partner of Gene Greenberg, who I worked for at Colorado. And so he gave me a ton of advice about where to apply and who to think about. And so I ended up doing my PhD in bacteriology at University of Wisconsin in Madison. And I have had a chance to look back and at my application materials, like the letter that I wrote, it was atrocious, and it said I was basically interested in everything. And that changed over the years, like in Madison, where I really got fascinated with metabolism and physiology, really where it stuck. And that then took me on to thinking more about, in my postdoc, I thinking about kind of environmental microbiology, or thinking about ecosystem microbiology, but still with the grounding in a system where we can do genetics and manipulate. And so for me, that like Shionella was a real natural fit. Like I didn't know Shionella, but I knew the molecular genetics, kind of the classic approach. I'm one of the few people that still knows how to like map point mutations with phage. Like we don't do that anymore because we can just sequence, right? But I brought kind of a lot of old school kind of thought to that system where some genetics had been done, but kind of not really with somebody with my background. And over the years in my lab, we've developed kind of more robust genetic methods as well, kind of more modern genetic methods. And sometimes that's easy to take something from E. coli and make it work in Shionella, but almost every time it's hard, even though they're both pretty closely related.
I know. Yeah.
Yeah.
And it's interesting because go ahead.
Oh, no, go ahead.
All I was going to say is that picking the right system is really critical. And at the same time, you've got to go where, what is it? The microbial heart wants what the microbial heart wants, right? So you're going to want to study what it is you want to study. And
Yeah.
sometimes it's not easy, but you can't
Now,
help yourself.
what I didn't know going into my faculty position is that Chiwanella put me in a really neat position to apply for funding almost everywhere. Now, that's not to say I've gotten funding from everywhere, because I certainly haven't. But like one of the grants that I wrote back in the day was like to the American Heart Association, because it turns out
Mhm.
Chiwanellas make these omega-3 fatty acids, which like concentrate in fast muscle tissue in animals and in fish. And like eukaryotes don't make that thing, but the bacteria do. So again, didn't get funded, but maybe someday we'll get back to that. So I have been fortunate to be able to kind of pitch this organism in lots of different ways, both from a fundamental understanding, but also through like the electrochemistry to these kind of more electrochemical applications, and an understanding of how microbes interface with these kind of insoluble or inert surfaces.
One of the things that happens, and this is why I asked about your route, is students will come to me, or people I'll see at meetings or just talking about stuff, and they'll say, in order to become a microbiologist, you have to do X, Y, and Z. And that's never been my experience. It wasn't my route. And so I like to ask microbial scientists like yourself about that, to demonstrate to listeners and viewers that, the expression, it's a big tent.
Yeah.
I would say that we all exist within a great big petri dish.
Yeah.
And there
totally.
there are many routes to that petri dish, and it's important for people to see that. I was completely uninterested in medical microbiology, right? And I'm still not interested in medical microbiology. It doesn't mean it's not important, and it's great for some people. When I say microbiology, that's what people usually think, but it isn't. And certainly not for me.
It reflects one sliver of the microbial world. But what I do like to point out, especially because a lot of the students that come into our college who are majoring in microbiology, but other majors too at University of Minnesota, they're pre-med, right? They come in, that's the career that they're most interested in. A lot of the methods that we use to ask questions and understand systems are the same. And one of the
Yes.
things I really love is that most of some of the most sophisticated methods that we use are kind of trialed and figured out in environmental microbiology, and then get applied in medical microbiology.
Yes.
And if people want to get into some of the details of what some of the most current interesting stuff is in that realm, just Google BONCAT, BONCAT, and you'll see this amazing work that was done in environmental ecosystems, and now that's being done in medical systems. That's a way to kind of tag active microbes in a more or less unbiased way. And it's really cool. And in fact, Roland Hatzenplicker would be a great person to get on your podcast someday if he hasn't already.
He has been, and he's a
Yes,
wonderful
good.
person.
Good.
I love the fact that he has a link on his email that tells you how to pronounce his name.
Yes, Roland. There are very few folks out in the world or in the petri dish with a morphology as Roland.
Yes. Yes. Well, I have to tell you, it's been a great session today. And I mean, I enjoyed it personally. I also like the way that you pitch things to people who are relatively new to thinking about microbes and people who have experience. I also love the historical aspects.
Thanks, Mark. It's been super fun. Yeah, I'm looking forward to hopefully hearing more of these in the future too.
Well, that's very kind of you to say. And I want to thank you. I want to wish you and yours the very best. And I will see you down the road, I hope. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, as always, with epic links beyond compare, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Jeff Gralnick can be found in the Department of Plant and Microbial Biology at the University of Minnesota in Minneapolis, Minnesota. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.